What are short tandem repeats, STRs? Have you ever wondered how scientists can identify individuals using just a small sample of DNA? One of the key players in this fascinating process is known as short tandem repeats. So, what exactly are these sequences? Short tandem repeats are specific patterns in DNA where a short sequence of nucleotides, typically ranging from 2 to 6 base pairs long, is repeated multiple times in a row. Imagine a sequence like CAGs appearing several times consecutively. That is a classic example of an STR. These repeats are scattered throughout the human genome and make up about 3% of it. What makes STRs particularly interesting is the fact that the number of times these sequences repeat can vary significantly from one person to another. This variability is what allows scientists to use STRs as unique genetic markers. When it comes to technology, analyzing STRs is often done using a method called polymerase chain reaction. This technique amplifies the regions of DNA that contain these repeats, making it easier to study them. The length of the resulting DNA product reflects how many times the repeat occurs. This method is much quicker and requires less DNA than older techniques that involved cutting DNA with restriction enzymes. In the realm of genetic engineering and DNA sequencing technologies, STR analysis is vital for various applications, including forensic science, genetic genealogy, and medical diagnostics. For example, STR testing can help diagnose specific genetic disorders caused by abnormal expansions of repeat sequences. Conditions like Huntington disease and Fragile X syndrome arise when the number of repeats in certain genes exceeds normal limits, which can disrupt gene function. While massively parallel sequencing, also known as next-generation sequencing, can analyze many genes at once, it currently has some limitations in accurately sizing all STR regions. However, advancements in long-read sequencing technologies are expected to improve the precision of STR analysis in whole genome sequencing data. Traditionally, STRs were thought to be non-coding regions of DNA, meaning they did not directly affect gene expression. However, recent research suggests that these repetitive sequences may play a role in regulating genes and could contribute to various traits in individuals. In summary, short tandem repeats are short, repeated sequences in DNA that vary among individuals. They serve as important tools in genetic identification, forensic analysis, and diagnosing certain genetic disorders. As sequencing technologies continue to advance, the ability to analyze STRs will only become more precise, linking this genetic feature closely with developments in biotechnology and computing.